Welcome back to another edition of the Virginia Cannabis Digest. I'm your host, Phil, and today we bring you Teaming with Microbes, Converted Notes. So, as many of you know, we have a lot of handwritten notes on different books, and we've been slowly typing them up and converting them. Before we get into it, I just want to give you a few first impressions that I had. So, today we're going to go over Teaming with Microbes by Jeff Lowenfels and Wayne Lewis. I figured we'd do it outside because, even though it's hot, got the hat on, we'll be okay. It's such a beautiful, bright day. And, we get our girl, Steph Cindy, in the background to take a peek at. So, this is exactly what it says. The Organic Gardener's Guide to the Soil Food Web. My first impressions of the book as I read it were, this is amazing. I mean, it gives you such an in-depth amount of information, practically at a college level, with not a lot of stuff that's being bogged down. I really liked that. The explanations that they used were very, very nice. They were not hard to follow at all. I feel like anybody can pick up this book and series in general and be able to follow it very easily. If I can do it, you can do it too. Part 1. The basic science. What is the soil food web, and why should gardeners care? Good soil has lots of life to it, not just on the surface and the first 4 inches of soil, but lots of microscopic life that combines to make a soil food web. Living organisms that you cannot see are bacteria, fungi, protozoa, and nematodes. A teaspoon of soil can contain 1 billion bacteria, several yards of fungi hyphae, several thousand protozoa, and a few dozen nematodes. Chemosynthesizers, bacteria that use energy from sulfur, nitrogen, or iron compounds. The rest of bacteria have to eat something with carbon on it. A great amount of photosynthesis is used by the plant to secrete exudates within the root system. The rhizosphere, the area of interaction between the surface of a plant root and the area surrounding it. Rhizospheres extend roughly 1/10 of an inch around the roots. It contains all the microscopic and bigger life that competes for exudates, water, or minerals. Root exudates are carbohydrates, including sugars and proteins. These wake up and attract specific beneficial bacteria and fungi living in the soil that subsist on exudates and the cellular material sloughed off as the root tips grow. Exudates attract bacteria and fungi that start attracting larger microbes, protozoa and nematodes, to eat them. Anything not eaten or absorbed is excreted as waste, which the plant's roots absorb as nutrients. They also help to control the numbers and types of bacteria and fungi within the rhizosphere. During the growing season, exudates assist in the waxing and waning of bacteria and fungi. This is based on the plant's nutrient needs and can be different types of exudates. Bacteria and fungi retain the nutrients they eat and carbon. Soil life provides the nutrients needed for plant life, and plants initiate and fuel the process by producing exudates. Soil life creates soil structure. Protozoa and nematodes are eaten by arthropods, animals with segmented bodies, joint appendages, and a hard outer exoskeleton. These are eaten by other larger animals, snakes, birds, etc., while each one of these creatures moves about the soil for each other or protection. They have an impact on the soil. Bacteria are small and attach themselves to stick to things or get washed away. To do this, they produce a slime which ultimately results in binding individual soil particles together. Fungal hyphae act the same but bind by making thread-like strings. Worms, insects, and anything that burrows moves through the soil, creating pathways for air and water. When a member of the soil food web dies, it leaves behind its nutrients for others, including plants, to use. Without this system, nutrients would wash away, but are instead locked up inside the soil food web bodies. Nutrients that are locked into the soil food web are immobilized. Healthy soil food webs control disease and other pathogens. Pathogenic bacteria and fungi exist, bud rot, powdery mildew, etc., and large and diverse soil food web will catch them and destroy them. If there isn't enough, or it is stronger, faster, and better suited for the current environment, the pathogens win. To control them, they do not usually hunt them, although this does occur. Instead, they compete with them for exudates, nutrients, air, water, and space. This keeps pathogens in check or even outcompeted to their deaths. Webs formed by beneficial mycorrhizal fungi can act as a barrier between the rhizosphere and the pathogens that may be formed in the surrounding area. Bacteria will coat surfaces so thoroughly that there isn't any room left over. When these numbers drop, the plant can easily be attacked. Mycorrhizal fungi establish themselves in a symbiotic relationship with roots, providing physical protection and nutrient delivery assistance. In return for exudates, mycorrhizal fungi provide water, nutrients, especially phosphorus, and protection. If the soil food web is not in balance, these fungi are eaten and the plant suffers. Bacteria can produce exudates of their own, and the slime they use to attach to surfaces traps pathogens. Bacteria and fungi can work in conjunction to form protective layers around roots in the rhizosphere and leaves in the phyllosphere. Leaves produce exudates the same way roots do, acting as a barrier to invasions. The role of the soil food web is to cycle down nutrients until they are temporarily immobilized by bacteria and fungi, and then being mineralized. Nitrogen is the most important nutrient. It is the basic building block of amino acids and therefore life. The biomass of each bacteria and fungi determines, for the most part, the amount of nitrogen readily available for plant use. The older the soil is, the more fungally dominated it is. Think old forests. The younger the soil is, the more bacterially dominated it is. Think about your annual gardens, such as cannabis. Studies show that agricultural soils have an F to B fungal to bacteria ratio of 1:1 or less. Forest soils have 10 times more fungi or more than bacteria. Plants prefer fungal, bacterial, or neutral soils. The path from bacterially to fungally dominated soils follow the same general path as plant succession. When organisms are eaten, most of the nitrogen is excreted as waste in a plant readily available, ammonium, which is NH4 positive. Depending on the soil environment, this is kept as ammonium NH4 positive or converted into nitrates by specialized bacteria. This conversion occurs when NH4 positive is released into bacterially dominated soils. It occurs because soils generally have alkaline pH and greatly increases the nitrogen-fixing bacteria to thrive. Fungi produce acids and lower the pH of the soil. This prevents nitrogen-fixing bacteria from thriving, and the conversions occur far less, if at all, which leaves the nitrogen in ammonium NH4 positive form. Chemical fertilizers provide nitrogen in the form of nitrates, which is NO3 negative. So we can see that there are a lot of different roles within the soil food web. And Chapter 1 basically introduces you to those roles. Those were the notes on that chapter, and now we're going to move on to Chapter 2, Classic soil science. Soil, all the loose, unconsolidated mineral and organic material in the upper layer of the Earth's crust. Weathering, the sum impact of all natural forces that decay rocks, physical, chemical, or biological. Wind, rain, snow, and cold physically break down rocks and begins the process of soil formation. Water freezing inside rocks increases its size by 9% and has the force of 2,000 pounds of pressure per square inch. Slimes and acids secreted from bacteria and fungi break down rock molecular bonds and is a common example of chemical weathering. Exposure to water, oxygen, and carbon dioxide also chemically weather rocks. Microbes also produce ammonia and nitric acids, which act as solvents. There are almost 90 different chemical elements in soil, and only 8 make up the majority: oxygen, silicon, aluminum, iron, magnesium, calcium, sodium, and potassium. Each has an electric charge on a molecular level, and in different combinations form electrically charged molecules that combine to form different minerals. Roots attaching themselves to rocks is how they biologically weather. Soil needs more than minerals to support plant life. Organic material that builds up from things dying and then being decayed is needed for conversion into humus, the end result of composting. Humus consists of very long, hard-to-break chains of carbon molecules with a large surface area containing electrical charges that attract and hold minerals. Minerals and organic matter make up the physical aspect of soil. Air and water make up the gaseous and liquid phases, which fills the voids in between the minerals and organic matter. Both can share a spot. Water moves in between the soil in 2 ways: by the pull of gravity or by the pull of individual water molecules to each other, also known as capillary action. Gravity pulls water down, bringing fresh air down to the roots where it is absorbed. Smaller soil pores contain a film of capillary water that is not influenced by gravity and is actually left behind after gravitational water passes through. The liquid is bonded together by the attraction of its molecules for each other, a force known as cohesion, and to surrounding soil surfaces, a force known as adhesion. This creates a surface soil tension, causing water to form a thick film on the particle surfaces. Capillary water can flow uphill and is a major source of water for plants. Hygroscopic water is a thin layer, only a few molecules thick, that is so thin that the bonds between soil and water molecules cannot be absorbed by the plants, but is critical for microbes to be able to move around. Even when it is dry, this layer exists. You would have to actually boil the soil to get rid of it. There are 3 different sizes of soil particles: sand, silt, and clay. All tiny particles carry electrical charges and are called ions. Positively charged ions are called cations, and negatively charged ions are called anions. Positive charges are attracted to negative ones. When a positive cation attaches to a negative anion, the cation is absorbed by the anion. Microorganisms in the soil are small enough to carry and be influenced by electrical charges. Sand particles are too large to carry electrical charges. Clay and humus have a lot of negatively charged anions that attract positive cations. Cations absorbed by clay and humus are calcium, Ca positive positive, potassium, K positive, sodium, Na positive, magnesium, Mg positive positive, iron, Fe positive, ammonium, NH4 positive, and hydrogen, H positive. Anions in the soil are chloride, Ci negative, nitrate, NO3 negative, sulfate, SO4 negative, and phosphate, PO4 negative. All cations and anions are plant nutrients. Cations in clay and humus are so strong that when a solution containing them comes into contact, the attraction is satiated and only about 1% of the cation nutrients remain in the solutions. Anions are repelled by the negative charge in the clay and humus and stay in solution instead of being absorbed. These are commonly missing from soils since it is easily leached away while watering or raining. Nothing is holding them onto the soil surfaces. Root hairs have their own electrical charges. When attached to soil, it can exchange its own cations for those attached to clay or humus particles and then absorb the cation nutrient involved. Roots use hydrogen cations H positive as their exchange currency, given one hydrogen cation up for every nutrient absorbed. Cationing exchange capacity is the sum of positively summed nutrient replacements that it can absorb per unit weight or volume, measured in milligram equivalents per 100 grams, or MEQ over 100 grams. High CEC numbers means that there are more nutrients it can hold and better for growing plants. Cation exchange capacity in soils depend on its texture. Sand and silt have far less, and clay and organic matter have more because they are already carry them. Every time a root tip exchanges a hydrogen cation for a nutrient cation, the concentration of hydrogen ions goes up, making the soil more acidic. Things usually balance out since plants also take up negatively charged anions using hydroxy OH negative anions as a medium of exchange. Adding hydroxy OH negative raises the pH since it is alkaline. Fungi and bacteria are small enough to have cations and anions and perform the same functions of CEC. So chapter 2, we're basically learning the different compositions of soil. You have your liquid, you have your water, you have organic materials and small mineral particles, and everything is layering together to work in a pattern that besuits the plants. Chapter 3, Bacteria. Bacteria come in 3 basic shapes in the soil food web. Coccus or spherical or oval, bacillus is rod-shaped, and spiral-shaped bacteria. These were produced by single-cell division in lab conditions. A single bacterium can produce into approximately 5 billion in 12 hours. Moisture is required for bacteria to uptake nutrients, release waste, move around, and transport enzymes they use to break down organic matter. When soils are too dry, many go dormant. They rarely die of old age, but usually by something else or environmental changes. They are primary decomposers, second only to fungi. Materials ingested by bacteria are held until it dies or it is consumed. Most bacteria prefer fresh, still green materials because their sugars are easier to digest than more complex carbon compounds, brown matter. They eat through the cell wall membranes. In active transport, proteins in the cell wall membrane act as molecular pumps, using energy to suck or push through their target through the cell wall, nutrients in, waste out. Different proteins transport different nutrients. Root exudates are certain bacteria's favorite foods. Anaerobic bacteria live in the absence of air. These types of bacteria foster pathogens and kill off beneficial bacteria. Aerobic bacteria require oxygen to thrive. Actinomycetes are different from other soil bacteria. They grow filaments similar to fungal hyphae. These are adept at decaying cellulose and chitin, too difficult to digest carbon compounds. These have also adapted to live in a wider range of pH than other bacteria. Cellulose is complex carbohydrate made up of long chains of carbon-based glucose and is the molecular material that gives plant structure. This makes up half of the mass of plant bodies. Specialized bacteria called cellulomonas carry cellulose breaking enzymes that release only when they come in contact with cellulose. Other bacteria release upon contact of everything, eating in a hit or miss manner. Most bacteria hit their limit of decay with non-carbohydrate lignin. Lignin is the tough component of barks and woody materials made up of more complicated chains of interlinked alcohols. Lignin is resistant to the enzymes produced by most bacteria and are left for fungi to decay. Bacteria in the soil food web play a crucial role in recycling three basic elements needed for life: carbon, sulfur, and nitrogen. The nitrogen cycle is propelled by specialized bacteria that convert nitrogen from the atmosphere into plant-available forms. This is required because atmosphere nitrogen is so tightly bonded, it becomes inert. In order to fix it, bacteria add either oxygen or hydrogen to the atmospheric nitrogen N2, producing either ammonium NH4 positive or nitrate NO3 negative or nitrite NO2 negative ions. This process is called nitrogen fixation. Some examples of bacteria that live in the soil and nitrogen fix are Azobacter, Azospirillum, and Clostridium. Rhizobium live in the root tissues of certain plants, such as legumes, forming visible nodules. Another part of the nitrogen cycle that starts in the soil involves the decomposition of proteins into ammonium NH4 positive. This is usually the waste product produced by protozoa and nematodes after eating fungi or bacteria. Special nitrate bacteria, Nitrosomonas spp, convert the ammonium compounds into nitrates NO2 negative. A second type of bacteria, Nitrobacter spp, convert the nitrites into nitrates NO3 negative. Nitrifying bacteria generally do not like acidic soils and diminish as the soil pH drops below 7.0. Bacteria slime has a pH of 7.0, and if enough bacteria are in the same area, the slime they produce keeps the pH in their vicinity above 7 and nitrification can occur. If not, the ammonium first produced by organisms in the soil is not at all converted to nitrate form. If the pH is 5 or lower, then very little, if any, ammonium is converted. Denitrifying bacteria convert nitrogen salts back to N2, which escapes into the atmosphere. They do not help in soil fertility, but are essential in that they keep the nitrogen cycle moving. Biofilms are bacterial slime that is a matrix of sugars, proteins, and DNA. This influences the pH where it matters the most in the rhizosphere, while buffering the soil in the area so the pH remains relatively consistent. Some bacteria can use their biofilms as a mean of transportation, literally squirting the substance out as a mean of propulsion. Biofilms save bacteria from the desiccation as the soil dries, often leaving a sticky globs of biofilms, and complete with an infrastructure of channels filled with water for transporting nutrients and waste. They can also be a defense against antibiotics produced by other organisms, including other bacteria. Bacteria colonies produced by slime are 1,000 times more resistant than individual bacteria to antibiotics and microbiocides. So we can see that bacteria are taking in tiny things, eating them, holding them in their bodies. When they die, they're dying in the area where the roots can readily absorb them. There are different types of them. They have different functions. Chapter 4. Archaea. Archaea are very similar to bacteria. They travel similarly, divide their cells in the same single-cell division, and have the same basic shapes with the addition of a few square-shaped ones. There are needle-like filament archaea that look similar to actinomycetes but have irregular lobe shapes, triangular or even similar to a teacup. Archaea cell walls hold other lipids, bacteria do not. It contains amino acids and sugars in the membrane that are different from bacteria. Archaea cell walls never contain chitin or any cellulose. They have evolved to be in harsh environments, geysers, tundras, deserts, etc., that bacteria would typically die in. They can use many substances for energy, such as sugars, ammonia, hydrogen, and metal ions. Archaea do not reproduce through spores, and they play a big role in the carbon cycle and can be photoautotropic, obtaining energy from light using CO2 as their source of carbon, chemoautotropic, using inorganic chemicals as their energy and CO2 as their course of action, photoheterotropic, obtaining light for energy and organic chemicals to obtain carbon, chemoheterotropic, using organic chemicals as both for energy and carbon sources. Archaea can survive extreme environments because they have developed specialized enzymes that work where hot or cold temperatures, and their proteins fold tightly to avoid destruction from salt, heat, or cold. When living in these harsh environments, they are anaerobic. They play a big role in the nitrogen cycle, but forming at depths bacteria cannot. Bacteria diminish and eventually fade out entirely the deeper you go. Archaeota are the most abundant ammonium oxidizers in soil. So we have a form of bacteria, basically, that is capable of going where anybody else cannot. Or when they get there and they're not suited for the environment, they have an ability to evolve slowly over time, which is how that happened. It's pretty cool. Chapter 5. Fungi. Fungi forms thread-like structures called hyphae that rarely close off so nutrients and liquids can flow through. A teaspoon of good garden soil has several yards of hyphae. These grow in length and create networks over a given area. Fungi produce phenyl oxidase, a strong enzyme that dissolves lignin. They are the primary decay agent in the soil food web. Branching out, they can obtain nutrients from multiple sources simultaneously. Once a nutrient is absorbed through its cell wall, it is transported back through the hyphal network, which ends on a root. This really assists when the fungi takes up commonly locked up nutrients such as phosphorus, since they can free up anything over time with their powerful enzymes. Some fungi trade nutrients for exudates, but most often nutrients are released as a waste after consumption by the fungi or when they die and decay. Much of what is released in nitrogen in ammonium form, NH4 positive, and nitrifying bacteria are present. This is then converted to nitrite, NO3 negative. Enzymes used by fungi are acidic and lower the pH below 7 the more that are present. Fungi also attract predator microbes and consume them in place, injecting enzymes and then slowly absorbing them. It uses the hyphae strands to trap them, swell up, and strangulate it within 1/10 of a second. Mycorrhizae fungi form symbiotic relationships with the plant. They bring in nutrients and water in return for exudates, and then become dependent on each other. Ectomycorrhizal fungi grow close to the surface of the roots and can form webs around them. These associate with hardwoods and conifers. Endomycorrhizal fungi penetrate and grow roots inside as well as extend outward into the soil. These are preferred by most vegetables, annuals, grasses, and shrubs, as well as softwood trees. Both types can extend the reach and surface area of roots. This has the potential to magnify the root's surface area by 700 to 1,000 times. This allows roots to get nutrients and water they would otherwise not been able to have reached and absorbed on their own. Mycorrhizal fungi can also do this across multiple plants at the same time. Endophytes are fungi that live most or all of its life inside plant tissue, and some grow in root tissue. Most trees and shrubs are infected by hundreds of different endophytic species, and all lab-tested plants so far have at least one kind within it. These fungi form asymptomatical relationships. So far, endophytes that have been tested offer resistances to insects and diseases for plants. Some endophytes cause plants to limit seed production. Most endophytic fungi are asomycetes or sac fungi because their spores are produced in a sac-like structure. Some endophytic fungi require specific plants as hosts, and others will infect many different plants. While an endophytic fungi may be just that for one plant, it can be pathogenic for another. Certain kinds of endophytic fungi produce toxins for aphids, other sucking insects, and even stop the grazing of mammals. Many endophytic fungi initiate the decay process as soon as the host plant dies. So we have different types of fungi, and they have different functions, but they all share a similar concept in that they are exchanging what they find around them for what the plant has to offer right then and there. And it just so happens that the exudates have everything that they need, and they have everything that the plant needs for the most part. Particularly the most difficult one, phosphorus, which we will learn is chemically locked up. Chapter 6. Algae and slime molds. Algae are single-celled, thread-like photosynthetic organisms such as seaweed and kelp. There are three kinds: marine, freshwater, and terrestrial. They require moist conditions to survive and are photoautotropic, which means they take energy from the sun and produce their own food. Similar to plants, they are primary producers and are not dependent on the soil's organic content or other members of the soil food web. Algae lack specialization that characterizes higher plants, as they have no true roots, stems, leaves, or a vascular system. They assist in binding soil particles with excretions of polysaccharides, mucilage, and slimes. Nematodes can consume them. Slime molds spend most of their time pursuing yeast and bacteria in the soil. These engulf their food sources and digest it internally. There are two groups of slime molds: Dictyosteliomycota, cellular slime molds, and Myzomycota, plasmodial slime molds. They have similar life cycles. Spores germinate into myzoamoebae, amoeboid organisms that live in the soil and ingest bacteria, fungi, and small protozoa, which lock nutrients up and prevents them from leaching out. They are food for insect larvae, worms, and beetles with specialized mandibles to cram it into their mouths. Myzoamoebae swarm together to make masses similar to a slug or dollop of jelly, or even sometimes vomit. So, evolution is funny. I guess something to evolve that is some food and helps bind with their secretions. You know, that's kind of cool. We don't want to lose any nutrients. By the time it got to 11, it started to get a little warm. Chapter 7. Protozoa. Protozoa are single-celled organisms with a nucleus, making them eukaryotic, domain eukarya with fungi. They are almost exclusively heterotrophs and obtain nutrients by eating bacteria, mostly sometimes fungi, and to a lesser extent other protozoa. Paramecia are common amongst them. There are 60,000 types of protozoa that live in water and soil. All require moisture to lead an active life. Protozoa come in three basic forms. Amoebae are single-celled animal pseudopods with amorphous forms. These are always on the move by pouring their cytoplasm, the soup with all of its life parts, into one or more false feet or fake appendages called pseudopodia, and have two types. One type has a shell-like exoskeleton and 5 predefined holes that pseudopodia appear at. The second type lacks any shell or predefined pseudopodia. These are usually large for amoebas, and many would be as visible as paramecia if they weren't so transparent. Amoeba lack mouths and ingest bacteria by surrounding them and engulfing them in gas bubbles that convert to enzymes. Whatever it is eating is absorbed, and then the waste is expelled. Ciliates are smaller than amoeboids. These use lots of rows of hairs to move around with. The hairs can make microcurrents that bring bacteria towards their mouths. Flagellates move with one or two long hairs. Protozoa form symbiotic relationships with bacteria. They are attracted to and enter an area where there is a good supply of bacteria. They can eat an average of 10,000 bacteria a day. Once bacteria become scarce, larger amoeba eat the smaller ciliates and flagellates and move on. This allows bacteria populations to return to soil-food web balance. Bacteria survive to repopulate by hiding in soil pores protozoa cannot reach, while also leaving behind their bacterial slime, making it more difficult to penetrate to the bacteria, which lacks the oxygen that protozoa require. Protozoa will also eat nematodes. Hygroscopic water allows them to live, travel, and reproduce. When there isn't enough water, protozoa go dormant, encasing themselves in a cyst. How long they can survive in a cyst varies from species to species. Some can survive several years. Protozoa are mineralizers and release nutrients that are readily available to plants. Approximately 80% of nitrogen a plant needs in the soil food web comes from protozoan waste. Bacteria and fungi are in the rhizosphere, and when a protozoa expels waste, it does it where the plant can absorb it. They assist in the decay process by inadvertently ingesting small pieces of organic matter, breaking them up, and making them easier for bacteria and fungi to absorb. Protozoa are prey themselves for some nematodes, and worms rely on them. Gardens will not have worms if there are no protozoa. Not all protozoans are beneficial, but these are kept in check with a healthy soil food web. So, we got some basically bigger microbes that eat the smaller microbes, and they make things readily available for the plant. There are a few different types of them, including ones that keep themselves in check so they don't overwhelm the soil food web balance. It's pretty cool. Chapter 8. Nematodes. Nematodes are non-segmented, blind roundworms that mineralize nutrients in fungi and bacteria. Although considerably larger than protozoa, they are still hard to see without a microscope. They are the second most dominant life form next to arthropods. A teaspoon roughly has 20 bacteria feeders, 20 fungi feeders, and 10 predatory and plant-eating nematodes. Their numbers will directly relate to the soil's fungal to bacteria ratio domination. Certain nematodes feed on the outside, ectoparasitic, roots, and others on the inside, endoparasitic, of the roots. They create lesions in the roots as well as cysts and large bubbles that are often referred to as root knots. These are not helpful to the plant. Bacteriivores and fungivores mineralize bacteria and fungi for the plants. Predator nematodes eat protozoa, algae, small members of the soil food web, and other nematodes, which keeps the destructive herbivores in check. Nematodes use less nitrogen than protozoa and release more nitrogen in the rhizosphere in ammonium form. They require porous soils to move around in. Compacted or the wrong texture soils reduces their numbers by dying off or moving away. This greatly diminishes the flow of nitrogen. Nematodes spread bacteria and fungi around because bacteria and fungi will attach itself to their outside and hitch a ride. Some fungi will attack them while the nematode is unaware doing its daily activities. Being blind, some nematodes find prey by soil temperature, heading to the proper temperature and eventually bump into your food source. Others sense particular chemicals associated with their food source and lock on to their prey this way. So you have a specific extra group of mineralizers that help account for quite a vast majority of nitrogen for plants. That's pretty cool. Chapter 9. Arthropods. Approximately 75% of all living creatures are arthropods. Still, the biomass of protozoa and nematodes is greater. Arthropods have segmented limbs, bodies, and all have an exoskeleton made from chitin. The head is a cephalon, the chest is a thorax, and finally the abdomen. Most arthropods live in 3 stages: eggs, larvae, and then metamorph into a very different adult form. Microarthropods require a microscope to see, and macroarthropods you can visibly see without aid. Besides being a food member for others in the soil food web, they play an important role as soil shredders, predators, and soil aerators. Arthropods constantly shred the soil, making smaller pieces, which makes it easier for fungi and bacteria to move in. Fungi and bacteria are also moved about from place to place as they hitch rides on arthropods. Mites and springtails are responsible for recycling 30% of leaves and woody debris on a temperate zone forest floor. When insufficient dead organic matter happens, they will attack living sources of organic nutrients. Some eat root hairs, root systems, and get inside the host plant. Others eat other members of the soil food web. Some also eat fungi, bacteria. These release nutrients in the same way protozoa and nematodes mineralize nutrients. Many arthropods carry out their routines on the soil surface. A surprising amount live at least part-time below the soil surface. The arthropods mix and aerate the soil, and their wastes add to the organic matter. There are 2 basic mites in the soil. Orbited mites have the highest populations of any soil arthropod, up to several 100,000 per square yard. A female orbited can lay eggs without a male. These inhabit soil surfaces, littered debris, living plants, and some feed on live nematodes and dead springtails. Most can eat fungi, algae, and decaying matter. Gossamer mites are major predators in the soil. They, as well, can have several 100,000 per square yard. These mites are entirely dependent on the availability of their food source, which happens to be any other arthropod in the soil. Gossamer mites that live in the soil also eat nematodes and fungi. So you have a handful of folks making sure that you're eating the other things and moving around, making sure things get decayed, more carbon's going back in, and more nutrition. Chapter 10. Earthworms. Earthworms shred organic matter, aerate the soil, aggregate soil particles, and move organic matter and microorganisms throughout the soil. They also increase microbial populations and aid in plant root growth. Worms eat bacteria primarily, but also eat fungi, nematodes, protozoa, and organic matter where these microorganisms live. Whatever a worm eats, it has strong muscles to break it down into particles, then it travels down to a crop and held until the gizzard is ready. The gizzard is a very strong muscle that has sand and small rock particles to grind its food up like teeth. After being ground up, it travels to its intestines, where it is mixed with a liquid calcium carbonate. Bacteria living in the intestines finishes the breakdown process quickly. Nutrients that are produced from the bacteria are then absorbed into the worm's bloodstream, and any organic matter not digested is expelled as waste. Vermicastings, worm poop, are 50% higher in organic matter than soil that has not been moved through worms. Bacteria in the worm's intestines is present in their castings and unlock many of the chemical bonds to make the nutrients more available. Castings are 7 times richer in phosphate, 5 times in nitrogen, 10 times in potash, 3 times in magnesium, and 1.5 times in calcium. All of these nutrients bind onto organic matter in fecal pellets. Vermicastins also increase this CEC because of the greater amount of charge holding surfaces. Worms produce 10 to 15 tons of castings on the surface annually. Worms shred material and burrow in the soil. While moving around, they often deposit bacterial populations where there was none. Some burrows are temporary and others are permanent. Roots use these for air, nutrients, and water. They can also spread nutrients several yards around. A noticeable worm population in the area is a clear sign of a healthy soil food web community. If these are here, then likely everything else is in order. So earthworms are pretty interesting. Don't really need to recap that, but they do a whole lot for you. Chapter 11. Gastropods. Slugs and snails are nocturnal. As they move around, they secrete a slime of glycoproteins, which is a sticky substance of sugars and proteins. Slimes harden to form a path recognizable to other slugs and snails, or the same slug returning from foraging. This slime also has chemicals to ward off predators if it is being followed. Slugs and snails primarily graze on organic matter, fungi, bacteria, and occasionally surface plants. They only spend 5 to 10% of their time above ground. These have a place in the soil food web as decomposers and by shredding their food to consume it. Their underground tunnels create pathways for air, water, and roots. The slime they leave helps bind soil particles. That's interesting. Those pesky little things that are always trying to eat your lettuce and whatnot, are actually fairly beneficial. I never knew that until I read that chapter. Chapter 12. Reptiles, mammals, and birds. All of these burrow and travel in or on the soil. This mixes, moves, and deposits organic matter into the soil. It also provides pathways and reservoirs for water and air. Their dung provides other members of the soil food web community nutrition that they break down into readily available nutrition for plants. They carry microbes on and in their bodies, spreading microbes from one location to another. It's really not a lot to get into about them. We're not interested in specific ones, just in general. So they are useful, but for us, I try to keep the animals out of the garden. Birds are very welcome. They are like aerial defense, like toads, frogs, that sort of stuff. Those are very welcomed. You can think of them as like your tanks. You can think of birds as like aerial defense, and they're just rolling through, just decimating. Part 2. Applying soil food web science to yard and garden care. Chapter 13. How the soil food web applies to gardening. Fully active soil food webs have better nutrition retention. Healthy soil food webs have improved soil structure that can create the right porosity, better water retention, drainage, and aeration, all necessary for healthy plant growth. They provide defenses for diseases and things that would throw the web off balance. Some members hunt enemies down, and others provide hormones and vitamins repairing damaged areas. Fungi and bacteria serve as barriers to plant and compete for the same resources a bad guy needs to survive: water, air, space, and nutrients. Soil food web members help manage pH where it counts the most, in the rhizosphere. This directly determines the type of nitrogen, ammonium or nitrate, in the area. Soil microbiology can even take care of pollutants. There are 19 general rules to following when using any soil food web. At the end of all of the notes will be the rules listed out as they are slowly introduced through Part 2. Rule number 1. Some plants prefer soils dominated by fungi, and others prefer soil dominated by bacteria. Plants need nitrogen to produce amino acids for growth and survival. When nematodes and protozoa consume fungi and bacteria, nitrogen is released as ammonium form in the waste stream. Ammonium is quickly oxidized and converted to nitrates by nitrogen-fixing bacteria. When they are present in sufficient numbers, this is almost always the case when soils are dominated by bacteria. Rule number 2. Most vegetables, annuals, and grasses prefer their nitrogen in nitrate form and do best in bacterially dominated soils. Rule number 3. Most trees, shrubs, and perennials prefer their nitrogen in ammonium form and do best in fungally dominated soils. So we're going to start seeing some introduction, and we're going to see how we're going to use it on this next few chapters. Chapter 14. What do your soil food webs look like? Different plants produce different exudates that attract different bacteria and fungi. These then attract varying parts of the rest of the soil food web. To see the health of a given area, dig a 12 by 12 inch hole and put the scoop dirt onto a tart to sift through and see what bigger animals are there. Record everything you see or don't see. Worms indicate at least a decent soil food web at work. A Burley's funnel can show you microarthropod populations. Sending the samples into a lab is the best idea for determining everything you need to know. That's pretty straightforward. You can do a little bit by hand. You could make a Burley's funnel by looking that up online yourself. Or you could, pay a lab to get a full comprehensive set of information of everything that is going on in your soil. Chapter 15. Tools for restoration and maintenance. There are 3 strategies to restore your soil food web. Applying the proper type of compost, mulching the right way, with the right kinds of organic matter, and applying actively aerated compost teas. Compost can inoculate an area and has fungi, bacteria, protozoa, nematodes, and organic matter. Finished compost never smells bad and smells earthy and fresh and has a rich dark coffee color. Mulches provide the proper environment for the soil's community organisms to live and eat. Mulch is a form of cold compost. Although it does not heat up like regular compost, it does decay over time. Different types of organic matter can be used as mulch to supplement different members of the soil food web. Actively aerated compost tea is a liquid easily extracted from compost. Air pumps are used to pump air into a mixture of compost, dechlorinated and otherwise non-chemical treated water, and microbial nutrients. Teas are easier to make and apply than compost. They have a higher concentration of microbes and can be sprayed on leaf surface where compost does not stick. Beneficial microbes in the tea outcompete pathogens for food and space. Frankia are nitrogen-fixing bacteria. Rhizobia can help inoculate roots of legumes and start producing nitrogen in as little as 2 weeks. Most of the phosphorus put into the soil becomes chemically locked up with calcium, magnesium, iron, and aluminum to form insoluble phosphates. In these forms, the phosphorus is not available to plants. Phosphorus solubilizing bacteria and fungi produce organic acids capable of converting it to a usable plant form. There seems to be a relationship between endomycorrhizal fungi and phosphorus solubilizing bacteria. Inoculation of phosphorus solubilizing bacteria can increase yields while reducing fertilizer need. Many rhizobacteria produce special metabolites including gibberellins, plant growth regulators, and cytokines. These promote cell division and greatly aid plant health and development. They also produce enzymes such as chitinases, breaks down chitin, and aids in nutrient uptake, solubilization of phosphorus, and can increase root branching. So we have 3 primary tools mulches, compost, and actively aerated compost teas. Chapter 16. Compost. Rule 4. Compost can be used to inoculate beneficial microbes and life into soils around your yard and introduce, maintain, or alter the soil food web in a particular area. Rule 5. Adding compost and its soil food web to the surface of a soil will inoculate the soil with the same food web. Depending on what is put into the compost, it can be made to be bacterially or fungally dominant, or an even fungal to bacteria ratio. This will determine the type of nitrogen available to the plants. Bacteria, fungi, and other microbes seek mostly carbon from organic matter. They also need some nitrogen to make enzymes used in the decay process and proteins needed to build structure and enzymes. Moisture helps provide microbes the proper environment and prevent them from going dormant or dying. Air is needed for the microbes that create the proper compost for your plants. Heat is required and does not come from the sun, but from the soil life's metabolic activity. This heat is what creates an environment that increases populations and causes them to change at the appropriate time during the compost cycle. Mixed correctly, the end is a rich, crumbly, dark, coffee-colored, sweet-smelling, humus soil that is full of life. Compost goes through 3 distinct temperature phases. The first phase, mesophilic, which has organisms that thrive in moderate temperatures between 68 and 104 degrees Fahrenheit. Work starts on difficult-to-digest chains of cellulose, which are broken into smaller chains of glucose, a process called depolymerization. Brown rot, fungi, and certain bacteria are active breaking down difficult-to-digest material. These produce endospores that are resistant to chemicals and heat, enabling survival through the next hotter phase of composting. They return after temperatures cool down. Larger soil organisms start to come in and assist in breaking apart organic matter and by depositing their microbial activity from their gut, resulting in further chemical breakdown. All of this metabolic activity creates heat, eventually hitting 104 degrees and becomes too hot for mesophilic organisms and the next set of organisms adapted to the heat to take over. The second phase of the compost cycle is the thermophilic phase, which can withstand temperatures from 104 to 150 degrees Fahrenheit and over. Complex carbohydrates are completely broken down, some proteins are decomposed, and hemicelluloses, more resistant structures, are decayed. Many more bacteria and fungi can join in or begin to play more prominent roles. All of this metabolic activity raises the temperature more, killing off pathogens that may be present. The first 2 stages happen rapidly, within 24 to 72 hours. If it is not heating up, turn the pile to add oxygen. Additional fresh green materials can aid if turning the pile didn't work. Compost piles are best to keep between 140 to 150 degrees for at least a few days to kill pathogens and weed seeds, which die at 150 degrees Fahrenheit. Compost piles should not go over 155 degrees Fahrenheit or it will burn off carbon. Turning a pile can also cool it down. Adding water and more carbon materials will also cool it down. Bacteria are the primary heat generators. Use a thermometer to check the temperature. As complex proteins and carbohydrates are broken down, the pile reduces metabolic activity and the temperature decreases. Mesophilic organisms take back over and the compost enters the maturation stage. Lignin is now completely decayed. Actinomycetes finish the process. These organisms also impact the fresh earth and smell of compost. Physical decomposers support the microbial population by causing populations of bacteria and fungi to increase. After the initial run up of the thermophilic phase, it is best to keep the compost pile between 104 to 131 degrees Fahrenheit. Keep the compost pile moist but not saturated during the entirety of the process. Compost is finished and mature when you cannot recognize the material it was made of. The ratio of carbon of nitrogen, C to N, has to be right to make compost ideally between 25 to 1 and 30 to 1. Too much carbon slows the decay process. Too much nitrogen can cause organisms to snatch it up, then the carbon is vented to the atmosphere or mixed with water and washed out of the pile. Correct ratios makes things go fast and the decay is complete. Brown materials contain carbon, which provides soil food web members energy for metabolism. Green materials are good sources of nitrogen. The fresher the material, the more nitrogen it has. Nitrogen provides soil food web organisms with building blocks for proteins and enzymes. Compost piles require at least 3.5 square feet in order to heat up properly. Rule number 6. Aged brown materials support fungi life, and fresh green materials support bacterial life. So, compost should never smell bad. It should always just smell like earth. It can take a little bit of time. It's going to take a little bit of work. There are some ratios we need to pay attention to. But in general, it's pretty easy. And we definitely don't want to let it dry out, because then our bacterial life is going to diminish and we're basically going to have to start the process all over again. Chapter 17. Mulch. Mulch is anything that can be placed on top of the soil to reduce evaporation, prevent weed growth, and insulate plants. It also helps against soil compaction from the rain and nutrients and shelter for certain organisms. When applying mulch, only do 2 or 3 inch thick layers and stay a few inches away from the base of plants to not rot them out. Rule number 7. Mulch laid on the surface supports bacteria. Rule number 8. Bacteria needs moisture or they will go dormant and die. Rule number 9. Mulches with less than 35% moisture are considered dry mulches. Fairly straightforward. Compost teas put the microbiology back into the soil. Compost teas can be applied to leaf surfaces where compost and mulch cannot. This helps the microbes on the phytosphere, and they can even help protect the plant from pathogens, outcompeting them for space. It is a fast and inexpensive way to manage soil microbiology. They are aerobic mixtures of beneficial aerobic microbes. Bacterial populations in a single teaspoon can range from 1 to 4 billion. It is made by suspending compost or vermicasting or both in an actively aerated container with dechlorinated non-chemical water and molasses unsulfured, as well as other nutrients depending on your needs, that is actively aerated for 1 to 2 days. Brewers used for this should be able to tease the microbes out but not kill them from being too strong. Biological lab tests can show you what is in it. If not buying a setup, a simple 5-gallon bucket with an aquarium pump, the biggest one you can get it for it, will work. If the tea smells bad, it is anaerobic and needs more air, add another pump. Do not use anaerobic teas. Keep the setup clean after each use by rinsing and wiping off remaining residue. Use a porous bag, paint strainer bags work well, over the aeration. We clamp it down to keep it in place. This prevents having to strain it out before applying it. Temperature is important when brewing microbes. If it is too cold, the activity slows, and if it is too hot, they are cooked off or go dormant. Room temperature is ideal, in the 70s. Get a small aquarium heater if it is too cold, and use clean ice if it is too hot. Make teas away from direct sunlight. UV rays kill microbes. Keep your tea maker in an area that can tolerate some spillage, some nutrient foam, and sometimes the aerator will splash if filled too much. Rule number 10. Sugars help bacteria grow, and kelp, humic, fulvic acids, and phosphate rock dust help fungi grow. Rule number 11. By choosing the compost you begin with and what nutrients you add to it, you can make teas that are heavily fungal, bacterially dominated, or balanced. Rule number 12. Compost teas are very sensitive to chlorine and preservatives in the water and ingredients. Vermicastings are a great substitute for compost. Molasses unsulfured at 2 tablespoons per 4 gallons of water will help establish bacterial dominance. Fish emulsions will support bacteria and fungal growth. You can add mycorrhizal fungi at the very end of the brew. Preferably use the tea within 24 hours and do not use anaerobic, bad-smelling tea. When using it as a foliar spray, do it before 10 a.m. or after 4 p.m. when the UV rays are weaker. Sprayers for foliar feeding must not exceed 70 psu or it will kill the microbes. It should be a fine mist and not splatting. Concrete sprayers are best, or backpack sprayers as well if affordable. So compost teas are kind of like not just like a single, it's like a Swiss Army tool. You can use it for most anything, but it's a little bit better than a Swiss Army tool because it's not a one-size-fits-all. You can quickly tailor it. That's pretty cool. So that's an easy way to maintain plant health. Chapter 19. Mycorrhizal fungi. The reason mycorrhizal fungi infect a root is because they want a continuous source of carbon, which is what exudates are. In return, they break down organic matter, unlock nutrients needed, gather water, and then transport it back to the plants. When a plant's root uses all of a nutrient, the root must grow into more, or if a mycorrhizal relationship is present, then it can be ferried in from longer distance that the plant is not at. They can also outcompete, thus killing pathogens. They can also outcompete, thus killing pathogens. Some can produce antibiotics in the right conditions. Plants fed mycorrhizal fungi develop healthy roots, stems, and leaves. Mycorrhizal fungi networks can feed multiple plants. They produce chelates, which breaks down the tight chemical bonds of inorganic matter. Then the nutrients are absorbed and delivered to the plant. Rule number 16. Most conifers and hardwood trees prefer ectomycorrhizal relationships. Rule number 17. Most annuals, vegetables, grasses, softwood trees, and perennials prefer endomycorrhizal relationships. Nitrogen uptake in either form is stimulated by the presence of mycorrhizal fungi. Some even have the necessary enzymes to make nitrogen out of protein from organic sources. Endomycorrhizae are responsible for as much as 80% of a plant's phosphorus needs. Mycorrhizal fungi produce and release phosphatases, enzymes that break off phosphate groups from molecules they are bound to. Phosphate is then absorbed and delivered to the plant. Studies show that if phosphate levels get too high, 50 to 80 parts per million, many plants will not form mycorrhiza. Once levels drop, an association can be established under natural system. There is no phosphate pollution. Ectomycorrhizae have several things in common. One, development of a fungal sheath or mantle around the outside of the infected root itself. This increases root surface area and holds water. It may also stimulate and increase root branching. 2, infected roots tend to be wider and shorter than those not associated with fungi. 3, there is a network of fungal hyphae, a hard-tick net, that grows in between the outer epidermal cells and the inner cortical cells. This is where the transfer of nutrients takes place. It can also act as a barrier of invasion from nematodes and other soil food-web microorganisms and pathogens. Ectomycorrhizae also can switch between living with a symbiotic partner and deriving nutrients saprophytically, getting carbon from decaying matter. Endomycorrhizae invade the cell walls of a plant, but not the cellular membrane. They have tree-like branching of their hyphae or arbuscule. Infected roots surround the arbuscule, which live for a few days to 2 weeks. After an arbuscule dies, the root cell returns to normal. These branching clusters of very small hyphae provide plenty of surface area for the transfer of nutrients Transfers occur without fungal protoplasm contacting plant protoplasm. They chemically trade at the cell wall border. Some produce storage structures known as vesicles. All are obligate symbionts. If there is no host plant, they die. Glomalin is the carbon holding superglue that coats the hyphae of AM, arbuscular mycorrhizae. This helps seal fungal hyphae, plugging gaps and preventing water liquid nutrient solutions from leaking while traveling through fungi to plant roots. Glycoproteins, sugar and proteins, make glomalin sticky. This helps bind soil particles together, improving soil structure. Mycorrhizal connections take time, dust seeds, seedlings, and transplants to help this process. So we got a brief overview of mycorrhizal fungi. He has a whole book on that, which is the third in his series. They are critical to surviving, truthfully, not just using less phosphorus, but they will help you survive a drought by getting water in times where it can't reach it. So if a plant is fighting for its life, it's not growing anymore. It doesn't have that opportunity. If it has something actively returning water to it, it all of a sudden can help keep cool, and now it can start growing again. Pretty awesome. Chapter 20, The Lawn. Follow the soil food web rules. Although we read this chapter, we did not find anything pertinent to what we do here on the Virginia Cannabis Digest. However, we highly encourage you read it and use it on yours, should you decide to own or already have the book written by Jeff Lowenfels and Wayne Lewis. Seriously, get the book. It's worth it. Get the book. They don't know me. I don't know them, but you should know this for sure. Alrighty. So that's basically the whole book. Here are the soil food web gardening rules listed out. 1. Some plants prefer soils dominated by fungi. Others prefer soils dominated by bacteria. 2. Most vegetables, annuals, and grasses prefer their nitrogen and nitrate form and do best in bacterially dominated soils. 3. Most trees, shrubs, and perennials prefer their nitrogen and ammonium form and do best in fungally dominated soils. 4. Compost can be used to inoculate beneficial microbes and life into soils around your yard and introduce, maintain, or alter the soil food web in a particular area. 5. Adding compost and its soil food web to the surface of the soil will inoculate the soil with the same food web. 6. Aged brown organic materials support fungi. Green organic materials support bacteria. 7. Mulch laid on the surface tends to support fungi. Mulch worked into the soil tends to support bacteria. 8. If you wet and grind mulch thoroughly, it speeds up bacterial colonization. 9. Coarse, drier material mulches support fungal activity. 10. Sugars help bacteria multiply, grow. Kelp, humic, and fulvic acids, and phosphate rock dust help fungi grow. 11. By choosing the compost you begin with and what nutrients you add to it, you can make teas that are heavily fungal, bacterially dominated, or balanced. 12. Compost teas are very sensitive to chlorine and preservatives in the brewing water and ingredients. Applications of synthetic fertilizers kill off or most of all the soil food web microbes. 14. Stay away from the additives that have high NPK ratios. 15. Follow any chemical spraying or soil drenching with an application of compost tea. 16. Most conifers and hardwood trees prefer forming mycorrhizal relationships with ectomycorrhizal fungi. 17. Most vegetables, annuals, grasses, shrubs, softwood trees, and perennials form mycorrhizal relationships with endomycorrhizal fungi. 18. Rototilling and excessive soil disturbance destroys or severely damages the soil food web. 19. Always mix endomycorrhizae with the seeds of annuals and vegetables at planting and or apply them to the roots at transplanting. All right, you made it. So the one thing that I wish was spent more time on, or mentioned, and I don't know if it was like, I would be surprised if there wasn't a lot of information at the time of publication. Original, I think it was around 2008 for this. See if I remember correctly. Close. Copyright 2010. So cover crop had to be known about by then. I'm sorry. I don't mean that in a disrespectful way. I'm just kind of, laughing at it. But cover crop is incredibly important option in the garden and in soil food life, especially when you are building new beds. That way you can help build a structure that maintains the soil from literal erosion. And as we've mentioned here in this video, you can form relationships with legumes and other plants that are actually going to make sure that there's nutrition left over in the soil. And as we've learned, 80% of that comes from certain types of relationships. So cover crop is very important, and it's also going to act as a mulch because, in a sense, because it's going to keep from the sun penetrating straight down to the soil. Now, it's not a perfect mulch, but it will act similarly to one. So it's pretty beneficial. And then when it's done growing, let's say it gets too thick. Let's say it gets too tall for what you're doing. You chop it and you drop it and you let it stay. It builds carbon, it drops the nutrients, and then it becomes the mulch that you're looking for. If you're doing that, you're probably doing that in an area where you're already doing annuals or bacterially dominated things, so it would make perfect sense to do that. So that's one thing that I wish was kind of on there. But my honest final thoughts about it, get the book. Get the book. If you don't have it, get the book. Read the series. You don't have to necessarily take the notes. We'll eventually put them out there for folks to obtain, as well as other notes. This was condensed down into 21 or so pages, I believe, and then this is, that's about 200 pages of reading. It's not terrible. It's pretty straightforward. Get the book. Let's just take a peek at the stud city that's been behind us this time. Oh, hey there, wasp. Doing your wasp thing. You hunt. Go hunt. So this is soil food web. I have these here so I can just untie it and get into it. That way, dog and whatnot has a harder time getting in through the netting, and I can still get in here. So, I don't know, so it's like, what, June 13th? This thing is like 5 to 6 feet We've got a lot more time left for vegging, and then, as you are all aware, double and tripling in size, depending on genetics. Never grown this one, so we'll see. So if you'd like that we did this outside, or if you would rather just have a voiceover and just a simple, serene background of a cannabis plant, put it in the comments. Let me know if you liked it or didn't like it. If you liked today's video, give it a like and subscribe for more awesome cannabis content. Thanks for watching, and happy growing, folks.